Mendelian ratios. How about something that isn't so easy something that's a pretty good prediction? What fraction of the offspring will be affected? We get a lot of families, line them all up. What fraction of the offspring? A quarter. Now that's a hard and fast prediction. 1/4 of the offspring are affected. When I have a mating between two homozygotes, so what am I going to do? I'm going to go out, I'm going to collect a lot of families. Maybe I'll collect 100 families because it'll be a particular disease, diastrophic dysplasia or something like that. Xeroderma pigmentosa, ataxia telangiectasia. And I will go to the disease foundation, and I will get all the pedigrees for all the families, and I'll see how many times there was 1 affected, 2 affected, 3 affected, et cetera. And on average, the proportion of affecteds will be 1/4. Except that it's not true. If I actually do that, I find that the ratio of affecteds is typically more like 1/3. It isn't 1/4. Now this should disturb you greatly because you know full well that m over plus by m over plus should give you 1/4 affecteds. But when you actually look at human families, it's not. Why? In other words, when we count up all the matings between heterozygotes, we'll collect all the matings that produce 1 affected child. We'll collect all the matings that produce 2 affected children. We'll collect all the matings that produce 3 affected children. But we will fail to collect those matings between homozygotes that produce 0 affected children. And so we will systematically overestimate the proportion. Of course, what we really have to do is go out and get all of those couples who were both carriers, but because they had a small number of children, didn't happen to have an affected child. That's not very easy to do, especially when you don't know the gene in advance. So when human geneticists try to go out and measure the 1/4 Mendelian ratio, you can't. But what you can do is the following. Conditional on the first child being affected. Now, what will be the proportion of subsequent children who are affected? 1/4. If I make it conditional, conditioning on having a first child who's affected, number 1 child who's affected, then I know I've got a mating between heterozygotes. Subsequent offspring now do not have that bias. And so human genetics, now in fact, you think this is a pretty cool thought, right? You got a condition on one. Turns out there's a very famous paper about cystic fibrosis where somebody forgot this point and made a huge big deal in the literature about the fact that 1/3 of the kids on average had cystic fibrosis in these families, and proposed all sorts of models about how cystic fibrosis might be advantageous and, lead to fertility increases and all that. In fact, it was just a failure to correct for this little statistical bias. Okay? This is what human geneticists do, is they've got to deal with a population. Now, there's one other trick that you can use to know that something is autosomal recessive. That trick is this. To cite this trick, I have to go back to a person called Archibald Garrod. Archibald Garrod was a physician in London around 1900. Garrod studied children with the trait alkoptonuria. Alkoptonuria was what alkopton means black, urea means urine. They had black urine. This was evident because their urine turned black on treatment with alkali. Who would treat urine with alkali? How would people know this? Sorry? Outhouses with lime. Yeah, and who's going to look at the children's urine or something like that? But you're on the right track. How about diapers? You wash diapers, cloth diapers, in alkali. They turn black. This was evident from black diapers. The kids' urine would turn black. So he observed this, and you know what Garrett noticed is when he studied children with alkoptonuria, he found that a very large fraction of affected offspring were in fact produced from matings of first cousins. Consanguineous matings. Now you laugh but in fact, consanguinity has been something that has been favored in many societies, and in Britain, particularly amongst the upper class in Britain in 1900, marriage of first cousins was quite common. But not as common as he observed. He found that 8 out of 17 of the alkoptonuria patients were the products of first cousin marriages. That's way off the charts because it's nearly a half, when in fact the typical rate in Britain might have been about 5%. So on the basis of that, in the early 1900s, Garrett was able to show, only a few years after the rediscovery of Mendel's work, that this property of recessive traits, enrichment in the offspring of consanguineous marriages, was a clear demonstration of Mendelian inheritance. Not only did he do that, but Garrett knew, because of work of some biochemists, and this is way cool, that the problem with the urine was that these patients put out in their urine a lot of what's called homogentisic acid. Homogentisic acid, HGA, which basically is a phenolic ring. What Garrett did was and that stuff turns black on exposure to air. What might produce from the things you've learned already some kind of a ring like that? What building blocks do you know have rings like that of things you've studied already? Phenylalanine, tyrosine, both have rings. Suppose somebody had problems breaking down homogentisic acid. Suppose there was some pathway where proteins were broken down into amino acids, including phenylalanine and tyrosine. And they were broken down into homogentisic acid, and they were broken down into I don't know what. And suppose, like we had up there, patients had a mutation in that enzyme. What would happen if I fed patients a lot of protein? In their urine, you would recover lots of homogentisic acid. Suppose I fed them a lot of tyrosine. I'd get a lot of homogentisic acid because the body couldn't break it down. Suppose I fed them a lot of phenylalanine. They would excrete a lot of homogentisic acid. Suppose I fed them homogentisic acid. I would get quantitative amounts of homogentisic acid. Garrett did this. These in the days before institutional review boards, informed consent, and that turns out it's harmless, feeding them proteins and things like that. But in fact, Garrett in 1911 worked out that this trait had to be recessive because of its population genetics, and inferred a biochemical pathway by feeding different things along the way, and was able to connect a mutation in a gene to a problem with a specific biochemical pathway. 1908. This was his Croonian lecture in 1908. 8 years after the rediscovery of Mendel, he's able to connect genetic defect, showing it's genetic by transmission, to biochemical defect, showing that he has a pathway that he can feed things into, and it all blocks up at the inability to metabolize homogentisic acid. He has connected gene to enzyme by 1908. What do you think the reaction to this was? Polite bewilderment, and it sunk like a stone. Nobody was prepared to hear this. This is very much like Mendel, in my opinion. Now, he was a distinguished professor. It was the Kroonian lecture. He got lots of accolades and all that, and people said, what a lovely lecture that was, and proceeded to completely forget this connection between genes and enzymes, genes and proteins. It was not till 40 years later or so that Beadle and Tatum, working with a fungus, actually neurospora, not yeast, demonstrated that all these mutants interfered with the ability to digest or to make particular amino acids, and wrote this up as the one gene, one enzyme hypothesis of how genes encode enzymes, and won the Nobel Prize for this work. But in fact, in their Nobel address, Beadle and Tatum noted, actually, you know, Garrett kind of knew all this, but people weren't ready yet to digest it. Genetics had just come along, biochemistry had just really been invented in the last 10 years, and the idea of uniting genetics and biochemistry was just something people weren't prepared for yet.